Hello, everyone. Welcome to the broadcast of our keynote presentation, Reinforcement Learning, Mechanisms and Implications. It is presented by Emad Eskandar, MD, Professor and Chairman in the Department of Neurological Surgery at the Albert Einstein College of Medicine at the Montefiore Medical Center. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Eskandar. I will now turn the presentation over to him.
Well, thank you for that wonderful introduction. And as you heard today, I'm going to be talking about reinforcement learning, its mechanisms, and potential implications. So we will start by reviewing the anatomy of the basal ganglia, which play a critical part in reinforcement learning. And it's important to recognize that there are, in fact, multiple parallel loops through the basal ganglia. There's the commonly studied motor loop, which is involved in Parkinson's disease, but there is also the executive and the motivational loop, which are involved in cognition and decision-making, respectively. And the basal ganglia can be thought of as first having a set of input nuclei, or the striatum collectively. That includes the putamen, the caudate, and the nucleus accumbens. And then output nuclei, the globus pallidus internus, and the substantia nigra pars reticulata. And for most of our discussion today, we're going to focus on the motor and executive pathways and the caudate and the nucleus accumbens. And that's illustrated here, where we have the various structures involved in the different loops, and we're going to be concentrating on the largely on the executive loop. So that includes the essentially the caudate, and we're also going to look some at the limbic loop, which involves the ventral striatum. Now, this area, the anterior striatum, is very interesting. So what you see on the left is a set of coronal images with staining demonstrating where the connections to different cortical areas are. And you can see that there's a very significant representation of broad areas of the frontal cortex in the striatum, including the dorso-lateral prefrontal cortex, the dorsal anterior cingulate cortex, and others. In addition, we all know that this area gets a very rich dopaminergic innervation, as you can see on the image on the right, the lower right. Now, dopamine is very important, and we tend to think of it as tonic dopamine levels in motor disorders like Parkinson's disease. But there's also phasic dopamine release related to reward, specifically whether there's an expected or an unexpected reward. So this is a figure from some work by Wolfram Schultz, where they performed recordings from dopaminergic neurons in the midbrain of an awake behaving primate that was performing a behavioral task. And initially, the animal pulls on the joystick and gets a juice reward. And if you can see in the upper histogram that there's phasic dopamine release directly after the animal gets the reward. Over time, that reward is paired with something like a visual stimulus or an auditory cue. And then you can see that the dopamine release now occurs at the time that the cue occurs, which anticipates the reward. And then interestingly, If on occasion you omit that reward, you actually see a drop-off in the dopamine release. And so this has led to the idea that dopamine signals a reward prediction error, where unexpectedly good outcomes lead to an increase in dopamine release. So that's a positive reward prediction error, and ultimately reinforcement of the behavior that led to that reward. Expected good outcomes lead to a steady state and a zero reward prediction error. And then unexpectedly bad outcomes have a negative prediction error and presumably would lead to a reduction in the behavior. There's also cellular models that fit in with this idea, where if you stimulate corticostriatal synapses, they can have a prolonged period of facilitation. So there's a cellular mechanism whereby the dopamine can actually get a prolonged facilitation that lasts up to 20 minutes. So kind of putting it all together is this idea of reinforcement learning. And you can sort of imagine this circuitry here where the cortex and the caudate are densely interconnected. And then the animals or person is sort of behaving. So let's imagine in this case it's a monkey, and the monkey sees a banana tree as illustrated up there on the left. And he climbs that tree and he finds that there are bananas there. And when that happens, initially it's unexpected. And so there's an increase in midbrain dopaminergic release, which then causes reinforcement of the particular subset of circuits that led to that behavior. And then over time, that behavior gets reinforced, and then the dopamine release sort of goes back to its baseline. So this is basically a simple way of understanding, the various things that we've seen so far. So based on this, we wanted to study some ideas, hypotheses. One, that the caudate nucleus plays a critical role in learning. Two, that dopamine is important in reinforcing associations. And three, that somehow modulating the dopamine release can lead to enhanced learning. Use awake behaving non-human primates, and this is a standard physiology setup. On the left, you see the instrument rack. On the upper right, you see where the animal sits and the screen that it's viewing. And on the lower right is a coronal MRI demonstrating the brain, and then the chamber place that allows access to all of the necessary areas. This is a basic task that we've used. This is an associative learning task. If you look on the left, this is the first screen that the animal sees. It's basically a central fixation point with 4 gray targets around it. And then a new stimulus comes up in the middle, something that the animal has never seen before. And then the fixation point changes color. Then the animal has to figure out which target to go to, essentially by trial and error. Let's say in this case, the animal chooses the right target, then the animal would get a tone indicating that it received the reward. Excuse me, and then ultimately an apple juice reward. So you can imagine there's 4 targets, so there are 4 new stimuli, one uniquely associated with each target. And then over time, the animal learns these 4 different associations, but it's not immediate. It takes multiple trials to get that. So this is an example of group data from recordings in the caudate nucleus of an awake behaving animal. And the graph in the center shows a number of things. The blue line shows the animal's learning curve. So the animal starts at about 25%, which is essentially chance. There's 4 targets, so 1 out of 4 targets means he's going to get it right about 25% of the time. And then as he starts to learn, it curves upward. There's a steep part of the learning curve where he's starting to make and consolidating the associations. And then finally, the curve sort of plateaus once he's mastered the association around 85%. So that's actually a group learning curve. And then the black line represents the group or population neuronal data from the caudate nucleus. And the most interesting thing about it is that it really peaks during the steep part of the learning curve, when the animal's actively making new associations. And that's approximated by taking the first derivative of the learning curve. So there's relatively little activity in the beginning when the animal's naive, and relatively little activity at the end when the animal's mastered the association, and the peak of activity occurs when the animal is actually learning. And this occurs primarily during the feedback period of the task. So once the animal has learned whether he has gotten the trial correct or not correct. Now we've taken this research further and looked at this activity in the caudate and the dorso-lateral prefrontal cortex, and these are single unit examples. And the colored lines represent different outcomes. So expected positive rewards, green is unexpected positive rewards, red is expected negative rewards, and blue is unexpected negative rewards. And what's interesting is that the neurons seem to represent unexpected positive rewards, which fits in with the reinforcement learning model. But they also, in some cases, respond to unexpected negative outcomes. So the right-hand graph shows a neuron that responds to unexpected positive rewards, and the right-hand one represents a response to unexpected negative rewards. And we actually found many neurons of both kinds in both structures, the caudate and the dorsolateral prefrontal cortex. So this table summarizes it, and it's a small table, it's hard to see, but the point is that there's actually a significant number of neurons that respond to unexpected negative outcomes, about the same percentage, 70%, as those that respond to unexpected positive outcomes. So it's pretty fairly evenly distributed, which again doesn't quite fit with the existing model. Now we've also looked at this in humans, and this is a somewhat different task. So in this case, we're actually recording in humans that are having deep brain stimulation surgery. And we're planning to put electrodes in the nucleus accumbens, or the ventral striatum. So on the right is an illustration of a patient being prepared for surgery. You can see the top of the patient's head, and you can see the video monitor there. And once we're doing the operation and we're doing microelectrode recordings for placement of the electrodes, and we perform the experiment. And the experiment is a gambling task, which is illustrated in the lower row there. So in this case, it starts with the patient fixating on a dot, and then the patient gets his or her card. And then the patient is forced to make a bet as to whether the computer's card is going to be higher or lower. And if the patient is correct, well, then the computer flips its card, and then depending on the outcome, they get a screen that says they won $20 or they lost $20. So it allows us an opportunity to see what happens with expected and unexpected outcomes. So in the middle upper panel there, you can see the patient's reaction times, and it basically follows what one would think. That is, the reaction times are quite fast for the 2 and the 10 card. So they're sort of no-brainers, if you will. You're always going to lose on a 2 and always going to win on a 10. And then the reaction times are a little bit longer for the 4, 6, and 8 cards, because they require some thought. And then on the right, you see the percentage of bets. So again, people perform in a reasonably rational manner. So most of the time they bet low on the 2 card, and then most of the time they bet high on the 10 card. So their behavior basically tracks what we expect it to do. Now, while they're doing this, we recorded neurons in the nucleus accumbens, and they also exhibited this response to unexpectedly positive outcomes. So as you can see in the upper panel, there's a big difference there between unexpected wins and then unexpected losses. And then expected wins and expected losses show very little effect. We didn't clearly see a big response to unexpected losses, unlike what we saw in the primate. And it's unclear if that has to do with the location being a different structure or the task, because it's not using actual food or juice, or it's just maybe a sampling issue because we had many fewer neurons in humans than we did in primates. But it's interesting nonetheless. And another interesting feature we found has to do with the 6 card. So the 6 card is right in the middle of the spectrum. Basically, there's a 50-50 chance of either winning or losing. And the patient can choose whichever way they want to go. And what we found is that the activity in the nucleus accumbens, which is illustrated in the lower left-hand panel, actually predicts what the subject is going to do about 2 seconds before they actually do it. So this is a really interesting finding, indicating that there's some disposition or sort of intuition that the subject has, and it's already manifest in the brain, a full 2 seconds before they actually perform the behavior. And so to summarize this bit about recordings from the human nucleus accumbens, the patients behave rationally. Single neurons encode unexpected wins and losses. Although the wins are more prominent, and that part is consistent with the reward prediction error. And interestingly, single neurons seem to encode the subject's expectation or intuition when the visual cue is neutral. Basically, there's an equal probability of winning or losing. So we wanted to investigate the role that dopamine might play into this, and particularly what happens with electrical stimulation, which is one of the ways we have to modulate dopaminergic function. So we use a technique called fixed potential amperometry. And if you can see in the right upper hand panel, there are 2 electrodes. There's a stimulating electrode and a carbon fiber electrode. And the carbon fiber electrode has its potential clamped at 0.8 volts, which is near the oxidation potential of dopamine. And then any dopamine molecules that are in the vicinity of the electrode can attach to it, and they become oxidized and donate 2 electrons, and that can be detected as a small current. So then we can look very specifically at the effects of stimulation and what it does to the local dopamine transmission. And as you can see in the lower panel, that with brief pulses of stimulation, in the white matter above the caudate, you don't see a dopaminergic current. And once you get in the body of the caudate, in the lower 2 traces, there is an actual dopaminergic current. And then next we explored the differences between high and low frequency stimulation. And this is best illustrated in the lower panel. So with low frequency stimulation at 20 hertz, you don't see much of a change in the dopaminergic current. There's a little increase with each stimulus pulse, and then it relaxes back to normal. If you go to 200 hertz, that's the black trace, see there's actually a significant increase in dopamine release that's sustained for the period of the stimulation, which in this case is 1 second. So this gives us a mechanism whereby we can potentially modulate the local phasic levels of dopamine and determine their effect on learning. And the real question is, can we have time stimulation that sort of mimics the natural dopamine release, and would that modulate learning in any way? So like I showed you previously, it's the same circuitry, and the idea is that the stimulation is modulating and enhancing the phasic dopamine release. So we actually did these kinds of experiments where the animals performed the same associative learning task that I showed you before, but then during the feedback period of correct trials, they got high frequency stimulation. So specific stimulation occurring at a certain time and after correct trials, or as a control after incorrect trials. So that's the basic paradigm. And at first we thought that this would somehow just disrupt learning or kind of screw it all up. But in fact, what we found is that this type of high frequency stimulation actually enhances learning. So here's a segment of the learning curve, and the control is in black. And then in blue is the learning curve with stimulation. And what you can see is that the learning is better. It's faster than occurs in the normal state. And then we looked at high frequency and low frequency stimulation. And we can only really see the effect with high frequency stimulation. So the blue line is higher than the black line. Low frequency stimulation, which is the red line, really didn't show the same effect. And interestingly, unstimulated trials in the same session didn't show an effect. So the effect wasn't sort of spilling over or leaking over onto other trial types. And then we looked at stimulation following correct and incorrect trials. So again, the baseline learning curve is in black. The blue line represents learning with stimulation after correct trials, and so we see the enhancement in learning. And then the red line shows what happens if we stimulate after incorrect trials. And here it's very interesting because it seems that stimulation after incorrect trials actually retards learning. It slows down learning. It's as if the animal has to sort of work its way around this inappropriate association to ultimately learn what needs to be learned. Since then, that was about 10 years ago, we've repeated this experiment and used combined stimulation in both the caudate nucleus and the nucleus accumbens, with the idea that the caudate nucleus is involved in learning and the nucleus accumbens is involved in motivation. And what we found was that combined stimulation actually leads to a very profound increase in learning. So if you look at the black line, that's the baseline. And then the green line is the stimulation in the caudate nucleus, and the red line is combined stimulation. And in this case, we really get a very significant effect, both in terms of the speed of learning and in the final performance. So not only do animals learn faster, but they become more accurate. So we found that this was very interesting, and this was recently published in 2016. So given what we've learned here so far, we can reasonably conclude that the caudate nucleus plays an important role in learning, that dopamine is potentially important in reinforcing associations, that stimulation after correct trials results in enhanced learning, but possibly due to enhanced dopamine release, whereas stimulation after incorrect trials seems to retard learning. And so this led us to the question of, could you use this type of stimulation to enhance the recovery or the learning after some type of brain injury? Like, could you use this in some viable clinical scenario? So to do this, we created a mouse model of traumatic brain injury. And this slide shows a histology slide of the mouse brain. And as you can see, the red arrow there shows the area that's been damaged by using a controlled cortical impact. And then this is the model that we then use to evaluate this potential treatment. And then the animals basically perform on a Morris water maze. So imagine that the animal has the TBI, the traumatic brain injury, and then they have an electrode implanted, and then they have to swim around this maze as shown in the upper right-hand side. So they swim from the edge to a platform that's hidden. They can't see it from the surface of the water. And there are visual cues on the sides that indicate to them where to go. And the two lower panels show deliberate electrolytic lesions that we made in the nucleus accumbens and the caudate nucleus to confirm that we were in the right place. And what we found is that there was indeed an enhancement in their learning on this water maze task. So these are different search patterns that the animals had with and without stimulation. So the dashed blue represents the cohort of animals that had no stimulation. And you can see that even by day 5, they're still sort of pretty lost and they're swimming all over the place. Whereas the red line represents animals that had stimulation after they reached the platform, and you can see they have much shorter and much more deliberate search paths. And this can be quantified as the escape latency, which is the time it takes them to find the hidden platform, which is very similar to the learning curve. It's just going in the opposite direction. So in this case, the black line indicates the normals, the controls, and they have a gradual improvement in their escape latency. The red line represents the injured animals that don't get stimulation, and so they do not perform well, and they continue to not perform well. And then the blue line represents animals that had a brain injury and had the stimulation. And you can see they have an improvement in their learning and start to approach the control animals. We also wanted to test if this effect was durable or if it was sort of evanescent. So in a subset of these animals, we stopped stimulation after 5 days, waited 10 days, and then tested them again at 15 days. And what you see is that there is a significant retention of the learned material. So the black and the blue lines, that is the control and the stimulated animals, essentially start at the same point. So it's not that they've forgotten or that it's evanescent. The effect is still persistent. And then ultimately the injured animals in the red line eventually start to get better, but they never get quite as good as the control animals or the animals that receive stimulation. We also tested this in humans. So as part of a separate project where we had electrodes implanted into the brain for the purposes of seizure localization, we had an opportunity to test 3 patients and to determine whether we could see a similar phenomenon. And these results are illustrated here. So this is the learning in 3 different human subjects. Panels A and B represent the learning curves where the electrodes were close to the caudate or in the caudate. And the blue line indicates the presence of stimulation, and the red line indicates that they were not stimulated. And so in both of these patients, there was indeed an effect of stimulation on learning as expected and predicted by the rodent and the primate studies. Panel C shows a subject in which the electrode was closer to the putamen, and there you don't see a learning effect. So it's very specific to the caudate. And this slide summarizes these findings that effectively we've seen this learning enhancement across 3 species now, starting with the mouse, where we use the Morris water maze, and then going to the non-human primate, where we've repeatedly shown this enhancement of learning, and then finally in a limited number of human subjects, but still saw the same findings. So this gives us a lot of confidence that, this is a real effect, particularly that we can find it across 3 species and using different behavioral tasks. So this kind of brings us to the end of the talk. And so, things that we can feel comfortable about are that the caudate and the dorso-lateral prefrontal cortex respond to unexpected positive outcomes consistent with the reward prediction error. Interestingly, though, caudate and dorsolateral prefrontal cortex neurons also respond to unexpected negative outcomes. And so the questions that are outstanding, for us are really what's behind this response to negative outcomes. Is it a drop-off in dopamine release? And that may be the case, but the signals seem to have equal amplitudes, and based on the dopaminergic signal, it doesn't look like there's enough bandwidth to get this negative response. So the dopaminergic neurons fire tonically at like 5 hertz. They can only go down to 0, but they can go up to 30 hertz. So it's not clear whether that's sufficient to represent this response to unexpected negative outcomes. So there are a number of other possibilities. It could be other neurotransmitters are at play. It could be somehow embedded in the circuit dynamics. But it's a very important thing because obviously learning requires both learning new associations and unlearning, forgetting, discarding useless or maladaptive associations. So to learn optimally, the subject needs to do both things well. And then in regards to the stimulation, we found that stimulation after correct trials leads to enhanced learning in the rodent, the primate, and the human. And so, the question is, could these be useful enhancing recovery after some type of brain injury, traumatic brain injury, stroke? And so we're hopeful that we can start evaluating this in a pilot trial in humans in the near future. And as I pointed out, one of the other interesting findings, though, is that stimulation after incorrect trials retards learning. It slows down learning. And so could you use some certain type of stimulation to mimic the response to unexpected negative outcomes? And more broadly, could you use that to weaken maladaptive associations? So on the one hand, we want to reinforce learning in brain-injured patients, but there are any number of conditions where somebody might have some persistent behaviors or associations that are not helping them. For instance, tics and Tourette's syndrome could be conceptualized as a maladaptive association. Similarly, obsessions and compulsions and obsessive compulsive disorder can be looked at as maladaptive associations. And for instance, triggers for smoking or other types of substance abuse can be looked at as maladaptive associations. And so the question is, can you use stimulation in a deliberate fashion to weaken these associations and possibly replace them with other more useful associations? So in my mind, those are some of the big questions, but there are, of course, many others to consider. So that concludes my talk, and thank you very much, and I look forward to answering any questions.
Thank you, Dr. Eskandar, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. First question is, how do you know the learning effect is not due to reward?
So that's a really good question. That is, how do you know that the effect isn't simply a perception on the subject's part that this is just an enhanced reward? And I didn't show this data, but we actually looked at this very carefully. So we, for instance, did experiments where the animal had a choice between getting juice or getting stimulation, and 90-some% of the time they selected the juice. We also did experiments where they had two conditions, one of just juice and one of juice plus stimulation, and they picked at about 50%. So that tells us that they don't perceive the stimulation as in any way rewarding or hedonic or positive. And it's well below the levels that have historically been used for, intracranial self-stimulation. So we're operating in possibly up to 1 milliamp, and those older studies have used all the way up to 10 milliamps. So we really don't think it's somehow a enhanced reward effect.
Well, thank you. The next one is, how specific is the learning effect?
This is a really good question, and I'll answer that in two parts. One is, we haven't really fully tested if this generalizes in any way. It seems relatively specific. And then the question is, where does this specificity arise? And I think it arises from the circuitry, kind of as I showed in the beginning. That is, for any given behavior, there's a subset of circuits that becomes activated, and it's that subset that then gets facilitated if the behavior leads to an unexpectedly good outcome. So the stimulation doesn't have to be very specific. It's the circuits that are specific. And I think that's how we achieve this effect. It's still to be determined whether it generalizes in some way to other related behaviors.
The next one, how durable is the learning enhancement?
This is also a really good question. And, the best answer we have for that is from the rodent experiments. And as you recall, in those experiments, we stopped stimulating and then evaluated the animals 10 days later, and the effect seemed to persist completely in that the stimulated animals and the controls started at the same place. And that's as long as we've looked, but that seems to suggest that 10 days would be certainly long enough to make a difference in someone's rehabilitation or learning.
I would like to once again thank Dr. Eskandar for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June 15th, 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.